Well, hey AP, hope you're doing well when you get this video. I'm sorry that I'm gonna sound a little rough in this. I've been dealing with a viral infection since Sunday, and my little man is sick, and he's might be in the room for a little bit, and we're just struggling a little bit. But it's all good. God is in charge, and He loves us. So I definitely want to make this video for you. Please check over the email, and yeah, we'll just take it day by day. Thank you for all your hard work and your determination. So I'm going to start off with chapter 6. It's all about metabolism, energy, ATP, and enzymes. I'm going to go over what I've already covered in class, just in case you wanted to review any of that, and then go over the two new sections that I want you to take notes on. And here we go. So we talked about metabolism, energy, ATP, and enzymes. And we discussed about how, bioluminescence and energy transformations and fireflies and how they make light. And we watched this video, and went to this content about how luciferase, an enzyme, catalyze the reaction, and it needs cellular energy, which is ATP, to catalyze that reaction. So here's some of the objectives that we discussed: different types of energy, thermodynamics, and all of that. So the totality of an organism's chemical reactions is called metabolism. And so it's property of life because we have to be able to build things and break things down. A metabolic pathway begins with a specific molecule and ends with another one. So for example, each reaction in this metabolic pathway is catalyzed by one enzyme, a specific enzyme to that reaction. And you start with A and your goal is product D, and then eventually we'll start learning about feedback mechanisms. And well, if you have a lot of C, could that be an inhibitor for A and all that? But I don't want to get too ahead of myself with that. I just wanted to let you know that understanding this picture right here would be very important, okay, like I said in class. So starting with A ends in D, but each reaction is catalyzed by a specific enzyme. And so catabolic breaks down. Like I told you in class, I like to think caterpillars eat leaves, or caterpillar construction equipment. And anabolic pathways are like, anabolic steroids builds, and I know y'all laughed at that picture, but anabolic steroids builds, and so there's different reactions, building and breaking. And so each reaction, chemical transformations occur in a series of intermediate reactions. Each reaction is catalyzed by a specific enzyme. And so metabolic pathways are similar in all organisms, which means, according to an evolutionary worldview, that we came from the same one cell. Conserve, whenever you see similar in biology or conserve, secular textbooks want you to think evolutionary biology, that we all came and evolved. Creationists believing in God creating the world would think differently. Same evidence, just a different worldview. And creationists would say like, oh yeah, we share similar metabolic pathways because we came from a similar designer. In eukaryotes, many metabolic pathways occur in specific organelles, and they can be inhibited, which means stopped, or activated. And so here's different forms of energy. For our purposes, kinetic energy is energy in motion, or it's mechanical movement, right? Potential energy can be stored energy, especially chemical energy, such as food. That would be very important when it comes to like ATP and various things. And so here's the flow of energy. Ultimately, all energy depends upon the sun. Good old solar energy right here. And so producers can create organic, which means carbon-based nutrients using the sun's energy. And so that is the foundation of food webs and ecosystems, and of course those famous ecology pyramids. Consumers cannot make their own food. They must rely on eating, which makes sense. First law of thermodynamics: energy cannot be created or destroyed, so the universe can't be eternal. So transformation happens. Food, chemical energy composed of organic molecules, proteins, carbohydrates and fats, all examples of chemical energy. And then each energy change produces some heat. Of course, that's unusable energy, which leads us to law of entropy. Every energy transformation makes the universe less organized and more chaotic. So entropy indicates the relative amount of disorganization. So nature doesn't like organization. Cars are only 20 to 30% efficient, and so it's most of the energy is lost as heat energy. And so here's organized, and here's disorganized, which means more stable because there's more entropy. And so this takes energy, and this, when it breaks down, it releases entropy, increases entropy, I should say. Here's more organized, potential energy, less entropy, and this is more chaotic, more movement on both sides. And so nature hates unequilibrium. Nature wants to have a balance on both sides. We can use that to do work eventually. First law of thermodynamics, energy is equal on both sides. Usable energy decreases as you go there. And we went through those questions. Okay, 6.2 is all about ATP. Free energy, which hopefully you watched the video on Bozeman Science. It's a great video taking those 5 notes. Free energy is the amount of energy available to perform work. So exergonic. So there's 2 different reactions, and again, my son's with me, so it might get a little loud, but exergonic reactions, products have less free energy. So it could be spontaneous under certain conditions, but it still probably needs an activation energy lowering by enzymes. Exergonic reactions have less free energy, and endergonic reactions, energy has to go into the products. So the products have more free energy, or the amount of energy available to do work, therefore it requires an energy input. And because of this, we can actually couple these reactions, such as this, right? So if A, and we went through this, right? So this happens spontaneously, A break it down to B and C, but D and E make an F, that's a positive delta G, which means F has more free energy than D and E. And so, but if we couple them, it's still energetically favorable. And so this happens a lot in biology, coupling reactions, especially with ATP. So ATP is broken down, you release that energy, and that can drive energonic reactions like active transport and movement and anabolic reactions. And here's just some pictures of different transformations of potential and kinetic energy and laws of thermodynamics. ATP is super important. It's the currency of energy. It's a famous example in biology. When cells need to do something that requires energy, they use ATP, much like when we need to use money to do things. Energy is released in the bonds between the phosphates, and so when ATP is hydrolyzed, hydrolysis reactions, breaking down with water, millions of molecules are done per second. And so here is the chemical formula of ATP. The energy is stored between those bonds. And so you can have ATP3, ADP, and then AMP, which is involved with CAMP, a signaling molecule, which we'll learn about down the road. It's only one, monophosphate. And so the more active an organism, the more ATP is needed. Its on hand is small. It's constantly being regenerated. So cellular respiration produces ATP from glucose, or from what we eat, not just from glucose, from various things. 39% of the energy form glucose is transferred to ATP. So a lot of energy is lost as heat energy. Why is ATP important? Well, it can be used in many different types of reactions. It's high enough to drive biological functions, and it releases 7.3 kilocalories a mole per energy. I wouldn't know that that value. I'm not going to test you on that. That's highly memorization based, but you do need to know why ATP is important and how it can be coupled to endergonic reactions to allow them to happen more easily. Okay, and here's that ATP cycle that you can see. Exergonic reaction breaks it down, right, releases energy. But then you have to build it back up, and so that has a positive delta G. Okay, and so ATP can drive building, right? And then free energy change is more energetically favorable when we're trying to build. So notice it's a positive energy change here. Won't happen, but we need this molecule. And so if we couple it with ATP, notice negative 7.3 is energetically favorable. ATP also drives active transport and movement. And so an example in anatomy is myosin assumes its resting state when it combines with ATP. So ATP kind of charges it, and then that allows you to contract. And that's the sliding filament theory in anatomy. And so negative delta G, it's going to run. When delta G equals 0, nothing's happening. So equilibrium. So nature is never at equilibrium. The metabolism as a whole is never at equilibrium. It's one of the defining features of life. I told you to start that when we talked about last time in class. Okay, so example, you keep breaking things down in small reactions. Negative delta G, negative delta G, negative delta G, which happens there. So here's some questions for that. All right, so let's go to 6.3 now. So if you see right here, here's, and I'm sorry for all these click to add title things. I'm just trying to make this for you. But here's lactose. I mean, we're breaking it down, lactase, right? Lactase. And we're bringing it down to galactose and glucose. And so this will be a metabolic reaction, right? And there's an example here, some data. I was going to go through this with you. Basically, it's an example of, oh, my bad. It's an example of different ways plasma galactose increases due to different cell lineages, but for time, we won't go into that. And so enzymes. Lactase is an enzyme. Some people don't make an active form when they're adults, and therefore they're lactose intolerant. Enzymes are crazy important in biology. Enzymes are protein structures, tertiary. Sometimes they also have an RNA component, but enzymes are crazy important. You need to know everything you can about enzymes. So protein molecules, they function as catalysts, which means they speed up reactions. They bind to substrates, and they usually just do one specific reaction. And each reaction in a metabolic pathway requires a unique and specific enzyme because form leads to function, right? And so each enzyme has a specific active site, which only a certain molecule will form and bind to that, and then it'll be able to catalyze that reaction. And so the end product will not be formed unless all enzymes are present and functional. And so this can lead to disease. Like, let's say you make 5 out of 6. You can't make G unless you have all 6. And so that is the foundation of many different diseases. Reactions usually occur in a sequence. So A is the initial reaction, and then you have intermediates, and then you have G as the final product. That is a metabolic pathway. And so catalysts are substances that speed up, and I'll speed up chemical reactions without, permanently altered, so they can be reused. Most biological catalysts are proteins. Proteins. Well, that's kind of cool. Maybe not. Anyways. And so an exocytic reaction releases free energy. Without a catalyst, the reaction will be very slow because there's an energy barrier between reactants and products. This is called activation energy, which you have to know about. And so activation energy, and let me see right here. We got right here. Activation energy puts reactants into a transitional state. So it's kind of seen like this in a picture. You have reactants, which are stable, and then you have your transition state, and then you have your product. So the delta G is negative, but you still have this transition state where the reaction takes place, and that's like a little speed bump, right? And so what happens is activation energy can come from heat, kinetic energy, but this would not work well in living systems because homeostasis, right, would get too hot. And so enzymes, what do enzymes do? Star this, highlighted, very important. Enzymes, well, let's just highlight it. Enzymes lower the activation energy, well, it's not good highlighted, by enabling reactants to come together and react more easily. So a molecule of sucrose, that's table sugar, in solution may break down in about 15 days. But with sucrase present, it occurs in like less than 1 second, right? And so here's a very important slide. I would know this, how enzymes lower the activation energy. So uncatalyzed versus catalyzed. Notice it's a lower activation energy, which increases the likelihood of that happening quickly. So energy of activation, molecules frequently do not react with one another unless they're activated. And so that's energy of activation. So one more time, enzymes operate by lowering the energy of activation, helping substrates react quicker. The active site, so the enzyme substrate complex. Here's an enzyme, kind of looks like a Pac-Man. The famous example is Pac-Man. And you have a substrate that binds specifically to the active site. Now remember, no brains here. We're just randomly moving around due to kinetic energy. Small, fast movements in microspace here. But the active site has a specific shape. And so when the substrate binds, the active site changes shape, and the shape forces the substrates together, initiating a bond. And so there's an induced fit model that says when an enzyme is induced to undergo a slight alteration to achieve optimum for the substrate. So it means it basically puts pressure and strain on it. So here's sucrase, and so when it binds, it puts like a little pressure on it, and then it breaks it down, right? And it lowers that activation energy. Enzymes use one or more mechanisms, so inducing strain, right? Bonds of the substrates are stretched, putting it in an unstable transitional state. Sometimes the substrates are brought together so that bonds can form quickly. Sometimes enzymes add chemical groups, like R groups, which are those various groups that we talked about, hydroxyls and carboxyls. You don't have to know those specifically for this class, but just knowing chemical groups can be added. An enzyme's 3D structures are so specific that they bind to only one or a few related substrates. Many enzymes change shape when the substrate binds. Okay, so this is called the induced fit model. So here's a computer example here. Here's the enzyme's active site. Again, this is a a protein, right? So we're talking like 3D tertiary structure here, alpha helices, beta sheets, and secondary structures. Of course, everything determined by the primary amino acid structure. And there you can see the enzyme changes shape, kind of closes the glove around the ball when you catch it in baseball, induced fit, and reactions take place. Some enzymes require ions or other molecules called cofactors. Vitamins and minerals, not zinc. Copper, iron, zinc, coenzymes are important because they can help activate or enable enzymes to work correctly. And so here are some examples of coenzymes. You don't have to know these, but just know that what you eat is important, right? How you live your life is important. Help maintain homeostasis and help you defend against disease, which, of course, I'm struggling with a little bit right now. Rate to catalyze reaction. There's usually less enzyme than substrate, so reaction rate levels fall off when all the enzymes are balanced. This is called saturation. So it basically means like, well, if all the enzymes are being used, then we can't speed up the reaction because no more enzyme is around. And so it looks like this, right? Reaction without enzyme increases steadily as substrate concentration increases, right? So we increase it. But reaction with enzyme is like crazy fast, right? But then it kind of levels out because all the enzymes are being used. So you got to add more enzyme to increase the reaction. Degradation, which is catabolic, we're breaking stuff down. We bind to a single substrate and then the substrate is broken down. Anabolic reactions, where of course we're binding to and building. And so here is degradation, bind the substrate, break it down to products. Here is synthesis or anabolic. We bind to and build one. So substrate concentration, enzyme activity increases. So these are just kind of factors of how enzymes are affected. So first off, enzyme activity increases with substrate concentration, which makes sense, only until the enzyme is saturated. Of course, more frequent collisions, again, kinetic energy. Enzyme activity increases with temperature, more kinetic energy, more collisions. Warmer temperatures cause more effective collisions. But of course, hot temperatures, so there is a range right here, right, will denature. Remember, shape determines function, right? So if you change the shape of the enzyme, you change the active site, which would therefore lower the reaction rate and possibly destroy the enzyme altogether. Okay, and so here's an example, right? There's the sweet spot or optimal range. And so by temperatures of ectothermic animals often limits rates of reaction. By temperatures of endothermic animals promotes rates of reaction. So there's just differences there. But usually most enzymes have a specific rate of reaction. Most enzymes also are optimized for a particular pH. Pepsin, trypsin. Which one do you think is in the stomach? Pepsin, right? Low pH, a lot of acid. Cells can regulate the presence or absence of an enzyme. So this is called regulation. This is a huge thing in biology, meaning we don't need to waste energy. We need to determine if we're going to be making an enzyme or not. So cells can regulate the concentration of an enzyme as well. So not only the concentration, the presence, and then even if the enzyme is made, we can activate or deactivate it. And this is highly important when it comes to regulation of products and gene products and various things. Of course, we already talked about this a little bit, but cofactors, molecules required to activate an enzyme. Sometimes they're non-protein organic molecules, so such as like these. Vitamins are important, right? B2 and various things. And so here's the thing. If you need the cofactor, the cofactor might bind and activate the active site to bind the substrate. Chemical inhibitors, natural inhibitors, artificial inhibitors can be used to treat diseases, kill pests, site enzymes. So here's an inhibition. Reversible, so this is very important. So how enzymes are inhibited. So let's say they're around, but we are stopping them from working. Reversible means the enzyme's not destroyed. And so here's your inhibitor. It decreases its activity. Okay? Not destroyed, inhibitor. Competitive, which means it binds to the exact same place as the substrate. Okay? They both bind to the same active site. Okay? So there's where the substrate binds. The inhibitor binds there. There you go. Sometimes, though, there's non-competitive inhibition. This will be very important throughout the year. When you make a molecule and it binds to an allosteric site, highlight this word, underline it. Allosteric is a different site than the active site. and it changes the enzyme shape. Notice that the shape is changed, and the substrate can no longer bind. So this inhibits it. It's still reversible. The enzyme's not destroyed, but it's non-competitive. Okay? So here's a metabolic pathway. Sometimes, though, the end product can affect enzyme 1. And so this would lead to negative feedback regulation. When we have enough of the product, we don't need to make more of it. Homeostasis, right? We don't want to waste energy. And so the end product actually binds to the allosteric side of an enzyme and blocks it from working. And so therefore we no longer make the end product when it's at a certain level. Okay? Reversible inhibition, non-competitive binds that we've already called this allosteric regulation. And so examples of this in science, penicillin acts by binding to an enzyme. It can't, the bacteria uses the enzyme to catalyze formation of crosslinks in its cell walls. And so if penicillin breaks this, therefore the cell bursts. Okay. Here's reversible inhibition again, competitive. Here's non-competitive at a different allosteric site, right? And so allosteric regulation, super important, guys. Can't underestimate how important this is. It'll come up in different topics. But allosteric regulation is not a non-substrate molecule binds at a site other than the active site. The enzyme changes shape, which alters the chemical affinity, right? Attraction of the active site for the substrate. And so it can activate or inactivate enzymes. So notice that it's not always just turning the enzyme off. Sometimes it can also be activating the enzyme. Okay? It can be modified, covalent binding of a molecule, such as phosphorylation, non-covalent, such as so on and so forth. But you can see right here, here's a substrate, active site's open. You add a protein kinase, a phosphate, so you activate it. Activator here, you activate it. So activation, allosteric could be activating as well. Phosphorylation is the adding of phosphates. And so here's a phosphate, right? But notice it was inactive here and active here. And so that is called phosphorylation. They're done, let's highlight this, very important. Phosphorylation is done by protein kinases. So phosphorylation can change various things, hydrophobic to hydrophilic. The enzyme twists and exposes the active site. So phosphatases, very important term, so you gotta know the difference here. Reverse the process by removing. Okay, so kinases add phosphates in a process called phosphorylation. Phosphatases remove phosphates and therefore reverse the process. Here's a picture, right? Kinase, phosphoprotein, active possibly. Phosphatase removes it. Okay. Metabolic pathways, the first reaction is the commitment step. The other reaction that happen in sequence. Feedback inhibition, which we talked about up here. I'll show you another picture as the final product acts as an inhibitor of the first enzyme or a random enzyme in the process. So for example here, you have this process where threonine is made into isoleucine, but if isoleucine is at a high enough concentration, it actually blocks one of the first enzymes. So that's feedback inhibition. And then there's something called irreversible inhibition. Irreversible inhibition is when the enzyme is covalent and binds to a side chain in the active site and it's permanently inactivated. It's basically destroyed. And so some insecticides work this way. Aspirin alone works this way. These proteins called COX-1 and COX-2, they increase inflammation in the body. I'm trying to help you heal, but if you take aspirin and stuff, it will block it permanently, irreversible. And so just kind of interesting there. DIPF is an insecticide, I believe, and so it's a toxin, I believe, neurotoxin, yeah. And so it blocks an enzyme called acetylcholinesterase, which basically prevents acetylcholine from getting different levels. I'm not going to go into that. It basically involves a neuromuscular junction. But yeah, it's covalent inhibition of the enzyme. Sorry guys, I'm trying. Cyanide acts as a competitive inhibitor to the enzyme side. So here's the electron transport chain making tons of ATP. Cyclone C is up here. Cyanide is super deadly because it stops making tons of ATP. And so enzyme inhibitors can spell death. Cyanide inhibits enzymes required for ATP. Sarin gas. There's a I think in the 90s there's like a sarin gas something in Tokyo maybe or something that was a terrorist attack or something, but it must become paralyzed. It inhibits an enzyme. Warfarin, blood pressure or blood clotting. Sorry. So warfarin thins the blood, but can cause internal bleeding. And then finally, we're almost there. Y'all done great. I hope you're doing great. Again, I'm sorry that I'm out. But here's the last one. Living cells require energy from outside sources. And so the puffin, this guy, obtain energy by eating other animals, right? But that's not the only way we can get stuff. And here's a picture. I think I gave it to you in your notes. But everything starts with light, right? And then photosynthesis makes those organic molecules, and then we take that to the mitochondria. And we make tons and tons of ATP, and the rest is released as heat. Notice that a lot is released as heat, the remainder of it. And so the last unit right here is basically about oxidation and reduction reactions, redox reactions. And so I want you to write this term here on your notes somewhere in a second. Oil rig, I'll show you in a picture in a second. But basically, oxidation is loss of an electron or hydrogen, and reduction is gain of an electron or hydrogen. They'll take place at the same time. And so these redox reactions, I don't know if you went over in chemistry, but I like to think of it as oil rig. Oxidation is loss, reduction is gain. So we're losing electrons and usually hydrogens, and we're gaining electrons and hydrogens. And if you're gaining hydrogens, you're gaining energy, burnable energy. And so the electron donor is called the reducing agent because it's going to reduce another molecule. The electron acceptor, now this is always confusing for students because it's like, biology vernacular here. But the electron acceptor is called the oxidizing agent because it oxidizes something else. Okay, so let's take this example right here. CH4, you can burn CH4, methane, tons of energy, hydrogens, right? It becomes oxidized. Oxygen becomes reduced, water. Okay, and so you can see right here, gaining hydrogens and electrons, losing hydrogens and electrons. But since whatever becomes oxidized is the reducing agent, because it was oxidized, it reduces something else. Whatever is reduced was the oxidizing agent because it accepted those donated electrons and hydrogens. So whatever is oxidized is the reducing agent because it donates what's necessary to be reduced. It's going to act as the reducing agent. Oxygen in this process is reduced to water, and since it is reduced, it is the oxidizing agent because it is taking or accepting the donation of those electrons and hydrogens. So carbon dioxide is reduced, right? So here's photosynthesis, right? So carbon dioxide is reduced to form glucose, and water is oxidized to form oxygen. So if carbon dioxide is reduced, then it is the oxidizing agent. If water is oxidized, it is the reducing agent. It's donating those hydrogens and electrons. And so here we go, right? This is the opposite. This is cellular respiration. So we take the sugar right here and we release those hydrogens. So it is oxidized. Thereby it is the reducing agents donating those electrons and hydrogens. And then oxygen is reduced. Thereby it is the oxidizing agent. Okay? Electron transport chain is very important in membrane-bound carrier proteins found in mitochondria and chloroplasts, physically arranged in an ordered series. So basically you have these high energy electrons. Electron transport chains happen in photosynthesis and cellular respiration. And so electrons have high potential energy, and we slowly extract that energy, we mine it to do work. And so electron energy used to pump hydrogen ions to one side of the membrane. It establishes an electrochemical gradient. So therefore it what that means, let me see if I can draw it with a mouse. But basically, electrochemical gradient is like, here's your membrane, and you have like tons of hydrogens over here. Oh man, that's not good. Zoom out. If you have tons of hydrogens over here, that's like a positive charge compared to negative or neutral charge up here. So that's like electrochemical gradient, tons of hydrogens. The electrochemical gradient is used to make ATP from ATP using a process called chemiosmosis. We'll learn more about that down the road. Chemiosmosis is a nice fancy word that basically is just meaning that we form something by letting hydrogens flow downhill, down a concentration gradient. So it looks like this. Hydrogen pumps, right? We're pumping all these hydrogens across, and then the hydrogens flow downhill, pump making ATP through ATP synthase. That's chemiosmosis, when you flow from high to low, forming ATP. Okay, so this video was a little bit longer than expected, but I hope it was helpful. Sorry again for all the noise and the coughing and my son playing with Legos, but hope you're doing well. So for the rest of class, let me go through a few things with you. And I mentioned this in the notes, but right here, so here's the end of chapter 6, at least online. Here's the topics. I would read over these and then do all these questions, see how you do. We'll review on Friday for sure. But here's that, and then check the email because I sent some other documents in the email, and some what I expect you to do today. Thank you so much for working hard, for listening to this. I hope you have a good rest of the day.